Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about energy and enzymes, two really important concepts as part of a larger picture, and that is talking about cellular metabolism. And so in today's video, our focus is really going to be on energy, the different types of energy, and enzymes, what they're made of, their properties, how we control them, and that will lead into a discussion later on about cellular metabolism. So let's go ahead and get started. We're going to be talking about energy, as I said. Now, energy is kind of like what we're seeing here, this water behind a dam. There's different types of energy out there, and we're going to relate this to cells later on in another video. But the two main types of energy we want to start out talking about today is the first is potential energy. And the reason why I have this picture here is all of that water behind this hydroelectric dam has potential energy. It's based on its location. And so this is, in essence, stored energy. Based on its location, it has the ability to turn into another type of energy, and that other type of energy is called kinetic energy, and that is the energy of motion. And so when we're looking at something like this, a hydroelectric dam, what we're seeing here is we have all this stored energy in the water behind the dam, and then a little bit lower, you can see that the water is coming through the dam, converting potential to kinetic energy. And the purpose of that is to spin a turbine that then produces electricity. And so that potential to kinetic energy can actually do work. And that is a really super important concept for us to understand. Now, when we are making that conversion, when we're actually having a reaction happen inside of a cell, some of that energy can actually be used to do work. And sometimes it could be the majority of the energy, and we call that free energy. And so free energy is the energy available to do work, and that is a percentage of the total energy released. A lot of times the rest of the energy is lost as heat to its surroundings, and that is the least usable form of energy. All right, so now that we have that background, we understand what potential energy is, energy based on location, kinetic energy, energy of motion. We have free energy, that is the energy that's available to do work. Let's see how this relates to an actual cell and a biological process. So now that we understand free energy, let's take a look at a couple other types of reactions. We're going to kind of go into a cell, if you will, and there's a couple different types of reactions that happen there. Endergonic and exergonic reactions, and there's a couple of key points that we want to understand here. And so let's start on the left-hand side with the endergonic reactions. With an endergonic reaction, you can see that we've got free energy on the left, and then down towards the bottom, we have time. Now, what's happening here in an endergonic reaction is the reactants, the things that are going to react with one another, they don't actually have a whole lot of free energy. But we add energy into the system, and we wind up with products that do have a lot of free energy. So let me give you an example on why this matters. A little bit later in a future video, we're going to be talking about the process of photosynthesis. And this is a really great example of an endergonic reaction in which you've got CO2 that really doesn't have a whole lot of available energy. Plants and algae and cyanobacteria, they use the energy from the sun along with some electron source like water, and they pack a bunch of energy into that CO2, and eventually you wind up with sugars like glucose. And the glucose has a lot of potential energy. Now, the important thing to understand with the endergonic reaction is the reactants don't have a lot of energy, but when you pack energy into the system, the products will. On the right-hand side, we have the exact opposite. We have an exergonic reaction. In an exergonic reaction, you can see here that the energy is actually quite high for the reactants. And then once the reaction takes place, you look at the products on the right-hand side, and wow, they don't have a whole lot of energy available. Now again, this is really important for us because in a future video, we're going to be talking about metabolism, especially energy metabolism when things like human beings, we take in glucose that has a whole lot of potential energy. And we break it down, and eventually we breathe out CO2 that doesn't have really hardly any energy at all, usable energy at all. And so it's the exact opposite of what we saw with the endergonic reaction. But the key point here is with an exergonic reaction, you start out with reactants that have a lot of energy, And then once the reaction is done, the products have a whole lot less. Now you'll see on this slide here that for the endergonic reaction on the left-hand side, they say, okay, it's not usually a spontaneous reaction because you're inputting energy into the system. And in a few minutes, we're going to talk about enzymes that are necessary for this reaction to take place as well. On the other side, the exergonic reaction, it is more likely to be a spontaneous reaction, although it doesn't have to be. And you still sometimes need enzymes to allow this to happen as well. Now, we can link these types of reactions with a couple of more terms, and that is anabolic and catabolic reactions. Many times with the endergonic reactions, another way to describe this is it's also an anabolic reaction. Now, these terms, anabolic and catabolic, are used to describe what's happening to the substances or the structures that are involved in the reaction. And so with an anabolic reaction, what's happening is we're building something. And so you're taking monomers, single units, and we learned about monomers in an earlier video. And so we're taking these single units, such as maybe we're building a protein and you've got amino acids, and you're combining those together to make a full protein. That would be an example of an anabolic reaction. So we're taking monomers and we're building them into polymers and we're making something. Again, usually, often that is going to be linked with an endergonic reaction because we're going to need an input of energy to do that. And when we're done building the thing, the product is going to have more potential or free energy than the reactants did. Again, on the flip side, we have the catabolic reaction. And in a catabolic reaction, we have the breakdown of something. And I explained earlier with the exergonic reaction, what we've got going on here is we have reactants that have a decent amount of potential or free energy. And then once the reaction takes place, we're left with something that doesn't have a whole lot of available energy. And the example that we used was breaking down glucose into CO2. That's a catabolic reaction. Breaking down often a polymer or a complex structure and breaking it down into its substructures or monomers. And so here what we have is a visual of what's going on in a catabolic and anabolic reaction. And so in the catabolic reaction on the left-hand side, we have a polymer. It could be a protein. It could be a polysaccharide, a larger sugar. And what's happening there is we're breaking that down, and we're breaking it down into its subunits. And as we do that, energy is being released. But here's the cool thing. As you release energy from a catabolic reaction, you can capture that energy and use it, as biological systems often do, to make things and take monomers in an anabolic reaction and bind them together into a larger structure. And so maybe we're taking amino acids and we're building a protein that the cell actually needs, or we're taking monosaccharides and building polysaccharides that the cell actually needs. So now that we know that, sometimes these reactions need a little bit of help to get started, and that is the role of enzymes inside of all of our cells. We have many, 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 many thousands of enzymes that are working to make reactions happen when they need to inside the cell. And in fact, a lot of the reactions that we need would not happen without the presence of enzymes. And so this is a depiction of how enzymes function and some things you should know about enzymes. Here at the top, this enzyme in green, there's going to be an active site, and that active site is going to interact with a substrate. And the substrate is the specific structure that interacts with an enzyme. And so you can see on the left-hand side, we have the enzyme that has the proper active site for the substrate, and then they are combined in the middle. And then in the top picture, we have an enzyme that is supposed to break that substrate apart, and then we have the products on the upper right-hand side. Enzymes can do the opposite as well. There are some enzymes that interact with substrates like you're seeing on the lower left-hand side. You have an enzyme substrate complex in the middle lower part, and that enzyme's job was to combine those two structures together to make a larger structure. So you could say that the top, the enzyme was actually performing a catabolic reaction, taking something larger and breaking it down. And at the bottom, the enzyme was going through an anabolic reaction where it was taking those structures and putting them together and building them up. All right, so now that we have the basics of how an enzyme functions, let's talk about its properties. One, enzymes are made of proteins. That's really important. 99.9% of enzymes are made of proteins. And the reason that matters is that when proteins are subjected to abnormal temperatures or pH or osmotic pressure, they can be denatured. They can fall apart. The next thing to consider is that enzymes can actually be reused, right? A single enzyme can be used to catalyze more than one reaction. However, they are very specific. Okay, so an enzyme that is going to function in some stage of glycolysis, breaking down glucose, is not going to be used to break down proteins or lipids or synthesize a reaction, right? So enzymes are specific. Really, really important. And this is a key for us to understand about enzymes. The way they work is by lowering activation energy. That is the amount of energy it takes to get a reaction started. That is a key point when it comes to enzymes. So remember that. Put that in your brain somewhere. Enzymes work by lowering activation energy. I'm going to show you a picture of that in a second so that you can get a better idea of what that means. Also, and this is a fun fact for you, from now on, when you see a word that ends in ASE, ACE, it's an enzyme, right? So when you see that lactase, galactase, a protease, lipase, when you see that ASE, that means, oh, I'm dealing with a protein, an enzyme that breaks down the thing that it is labeled. So let's talk a little bit more about how these enzymes are functioning. I said they lower activation energy, or the energy of activation means the same thing. So here again, we're looking at this figure where we've got energy on the left-hand side, we have time down at the bottom there on the X. And what we're seeing here is we've got some reactants. That would be the bluish purple line all the way to the left. And so we've got our reactants here, and it's showing you two different scenarios. The blue line is showing you the amount of energy it takes to get a reaction started. That is our activation energy. So activation energy, the amount of energy it takes to get a reaction started. It's kind of like a little bit of an assistant, right? A motivational speaker, if you will. And so without the enzyme, you can see that a particular reaction might have a really hard time getting started. But with an enzyme, and that's that purplish pink line, there is less energy required to get the reaction started. And so again, we're saying we're reducing the activation energy, or lowering the activation energy. And then again, at the end over here on the right-hand side, we've got the products. Now here's an interesting thing. If we're kind of putting together what we've learned so far, if we're looking at this, my question to you is, based on what we're seeing here, so we've got the reactants on the left-hand side, we have the products on the lower right-hand side, would you say that this is an exergonic or endergonic reaction? Now, if you said, oh, you know what? This sure does look like an exergonic reaction, you'd be right, because the reactants on the right-hand side had more potential energy than the products did on the right-hand side. So the reactants on the left, higher energy, products on the right, lower energy. Okay, so we've got these enzymes. Now, really important, we can't just have enzymes doing whatever they want, however they want. We have to be able to control them. And so often, one of the ways in which enzymes function is with a little bit of help. And so sometimes enzymes need coenzymes or cofactors to help the substrate fit into the active site. And that's what this picture here is showing us. And so we have this big blue enzyme here on the left-hand side. And then you can see here we have a coenzyme that is going to help the substrate fit nice and tightly into the active site. Now, generally speaking, the difference between a coenzyme and a cofactor, cofactors are inorganic. Coenzymes are organic, which means they are carbon-based. Either way, what we see here is that we have an assistant. So when you see coenzyme, cofactor, you're thinking, okay, this is an assistant that's going to help the enzyme bind to the substrate the way it's supposed to. Now, once again, just keep in mind that enzymes have a specific set of environmental parameters that they will function in. And so here is a great depiction of how enzymes function at different temperatures. Now, this happens to be an enzyme that has an optimal rate or an optimum temperature in the upper 40 degrees Celsius. So this would actually be an enzyme that's probably not going to function the best at human temperatures, but maybe in an organism that has a higher temperature than we do. But we can see a couple of important things here. We see that green line. We see that the rate of the reaction, that's on the y-axis, and again, temperature on the x, what we see here with that green line is that as the temperature increases, the temperature of the surroundings increase, that enzyme activity is also increasing. Now you see the red vertical line there, we see the optimal temperature, optimal rate. In this particular picture, that's somewhere around 47 degrees Celsius. Again, human body, we're right around 37. So this particular example, the optimum temperature for this enzyme is right around, well, let's say 47 degrees. But now notice what happens. Once we go beyond that optimum temperature, the protein denatures. It falls apart very quickly. And that is very characteristic of a lot of proteins. Once you take them beyond their temperature, they start to fall apart, they start to denature. That is the same thing that happens with pH, and it can happen with osmotic pressure, the amount of salt or solute in the surroundings. Now for pH and osmotic pressure, there might be a little bit wider range. It might not fall off as drastically, but there is still going to be a decrease in enzyme activity when the environmental parameters for that enzyme are not ideal. Now this next picture here just kind of shows you, hey, here's an example of what can happen. On the left-hand side, we have a depiction of a protein. And we can see here we've got some beta-pleated sheets, that's the arrows, and we've got some alpha helices, those are little corkscrew-looking things. But what happens is maybe we have an increase in temperature like we just saw, we have a pH that is less than ideal, we have osmotic pressure that's way too high or too low, and we wind up with a denatured protein. In other words, what's happened here is that protein, once it falls apart, it can no longer do its job because the three-dimensional structure of a protein is really important, just like we've talked about in an earlier video. And so what we really need is for that protein to be in its proper three-dimensional structure so that it can function. Once it's denatured, it's not going to work. Now, here we're showing an arrow that can go back into the renaturation of the protein. Now, in a biological system, that can be pretty tough to do. Sometimes when the temperatures or the pH or the osmotic pressure returns back to normal, you might have a protein, provided that's not too heavily damaged, it may be able to function once again. Okay, so now that we have that, let's talk a little bit about how we control these enzymes, because it's really important that we control enzymes and their function. And so the first way that enzymes can be controlled is through a process called competitive inhibition. And the term sounds just like its meaning. What we're saying here is that we can have an inhibitor, and that is the greenish blob in the upper left-hand corner. We have the enzyme in the lower right-hand corner, and you can see it has an active site. That's the site where the substrate binds. We learned that earlier in the video. The inhibitor is going to compete with the substrate for the active site. And that's the competitive inhibition nature of what's going on. So inhibitor and substrate, the yellow ball in the upper right-hand corner, they're competing for that active site. Whichever one gets there first wins the race, so to speak. And so if the inhibitor gets there first, the substrate cannot bind and we don't have a reaction. That can be a really useful thing, right? Inhibitors are part of a healthy biological system. We need inhibitors to stop enzymes from functioning when there's a feedback mechanism that tells the cell, hey, we have enough of the product. We don't need to be converting any more substrate to products right now. And so this is an example, a simple example of inhibitor, substrate competing for the same site. Now, if the inhibitor releases, the substrate now has the access to the active site, we'll get some more products. Now, there is another way in which enzymes can be controlled, and that is through a process called non-competitive inhibition, and that's on the bottom middle part, non-competitive inhibition. And again, just like the term implies, we have an inhibitor that is not competing with the substrate for the active site, but its end goal, when it does bind to the enzyme, is still the same. It still stops the enzyme from converting substrate into products. And so what's happening here is you've got this red inhibitor, and it's binding to some other area. And so in non-competitive inhibition, another way to say that is allosteric inhibition. So in non-competitive or allosteric inhibition, you have an inhibitor that's binding to the enzyme in a site that is different from the active site. Now, I mentioned here there's another term that we use, feedback inhibition, when it comes to a type of non-competitive inhibition. And so when you're organizing this in your brain, we talked about competitive inhibition in the previous slide. That's kind of a standalone sort of thing. But in non-competitive inhibition, we really have three terms that we're using at this point. So non-competitive inhibition is what we're looking at here, which we can also call allosteric inhibition. But there's a more specific type of non-competitive inhibition. That's called feedback inhibition. And the only difference there, really, for us to keep it simple, is in feedback inhibition, that red inhibitor, we're now defining what that is. And that red inhibitor is actually a product from one of the reactions that are happening downstream of the reaction we're looking at here. So imagine for a moment that you have an assembly line and you're assembling a car or a pen. It doesn't really matter, but you're assembling something. And there are multiple steps. There are multiple steps to make the finished product. Well, what happens when you've got enough of the finished product? How do you tell the workers or the enzymes earlier in the process on that assembly line, hey, we're done, stop doing things because we have enough of the product? Well, what happens is the product, the finished product at the end of the reaction stream, comes up to one of the enzymes earlier, maybe the first enzyme in the reaction on that assembly line, and says, boom, I'm going to bind to you, I'm going to shut you down. And now that the first enzyme doesn't have anything to do because it's bound by feedback inhibition, guess what? It can't convert its substrate to a product that enzyme 2 needs. And so now automatically, once you shut down enzyme number 1 through feedback inhibition, all the rest of the enzymes in that reaction process don't have a whole lot going on. All right, so that was a brief overview on energy and enzymes, which we need because in the next video, we're going to be talking about energy metabolism. We're going to be talking about ATP and glycolysis and how our cells use this vital source of energy to power all the reactions inside of our cells. As always, thank you so much for being here. If you want to see more on that metabolism, check out the video in the upper right-hand corner. Have a great week, everybody.